Hello, everyone. My name is Amaya Weddle, and I'm going to be moderating today's session. This is session 28 of talks at the 7th Polygen Symposium of Rising Scholars. So the goal of this symposium is to celebrate and showcase the hard work of our Polygen scholars, all of whom have tirelessly worked on their projects over the last couple of months. Today, we're going to have 4 students presenting on a variety of topics, and a fellow member of the Polygens team will be scoring each student's presentation, which will help us determine prize winners after the event. So during the sessions, if you have questions for our speakers, please type them into the Q&A, which is a box that you can find along the bottom of the Zoom screen. And we're going to have, after all the scholars have presented, we're going to have a Q&A session that I'll help to moderate. So I'll be checking that list. And then I also have a variety of questions myself that I'd like to ask. So today's speakers are Nambita Sahal, Alyson Nguyen, Saanvi, Lavande, and Angie Cheng. So without further ado, I'd love to introduce Nambita, and I will give you the floor.
I'll share my screen. Okay, so hello everybody. Today I'm going to be talking about a deep learning approach to categorize and estimate evolutionary conservation for the protein soluble guanylyl cyclase. So without further ado, by using biomimetic and evolutionary mechanisms, I have studied pharmacology applied to vasodilation and secondary signaling molecules. Through this presentation, I will be going over new technologies such as AlphaFold and molecular dynamics, as well as research I did in comparing species to determine the functional properties of protein. So major depressive disorder is characterized by depressed mood, impaired cognitive function, and MDD affects approximately 7.1% of the US population. Additionally, moya moya is a condition in which the blood vessels that supply blood to the brain become narrowed. This limits the flow of blood to the brain and puts them at risk for stroke. Moya moya and MDD have both been shown to be caused through mutations in soluble guanylyl cyclase. The SGC receptor has been shown to greatly increase risk for these disorders, but has a plethora of unknowns, including the signal transduction mechanism, the number of nitrogen oxide molecules needed to activate it, and more. By targeting the soluble guanylyl cyclase receptor as a site in which GTP is converted into cGMP, we can determine the fit of pharmacological properties for treatments that will work well with this protein. So, as I mentioned before, there are very few to no therapeutics targeting SGC, this cGMP pathway. And the study first categorizes the protein through models created using deep learning software. And SGC is also the start of a signaling cascade, which leads to an increase of vasodilation in blood vessels and helping blood flow. And our goal is to find structural information and then find drug candidates after that. So by first determining the structure of the proteins and then the binding affinities of drug candidates, and we can apply AI and machine modeling such as AlphaFold, we are able to better understand the structure and function of this protein. So to start off with this protein homology modeling, AlphaFold predicts a protein's 3D structure from an amino acid sequence. It can take in data from UniProt and output a prediction with accuracy. So we want to ensure that the model is accurate in the HNOX and catalytic domain specifically, as this is where a lot of the interactions with other proteins and treatments will be taking place, or at least hypothesized to take place. So the AlphaFold results do display confidence scores, which do not interfere heavily with the HNOX domain, which is where heme binds and a lot of important structures will bind. And there's a visual depiction on the left and right. In this case, the dark blue would demonstrate a high confidence, and we can see there are only a very few parts with low confidence, which can be shown through this orange light color. And after visualizing our model through alpha.
Hi, sorry, Nambita, I'm sorry to interrupt you. I'm having trouble seeing your screen. I was wondering if you could just try resharing and.
Oh, okay. Is it visible now?
It's still black. Are you sharing through the application or through like your overall desktop screen?
I believe I'm doing it through the application itself.
If it's okay with you, maybe we could try doing through the desktop and we might get a different result because it's just showing black.
I can do the desktop. Is it visible now?
No, I'm sorry, it's not showing for some reason. It was working, of course, when we were practicing, but let me see. Would it be okay if you just spoke verbally?
Yeah.
I was following along with what you were saying, but
okay, I can speak verbally. I think some figures, I can just explain the figures through the descriptions. Yeah.
Sounds great. Okay, sorry, continue on.
Yeah, after visualizing our models through AlphaFold, we needed to look at conserved residues of the protein. What that means is that these residues are structurally sound across multiple species for the protein. So if you see similarities with many species, then that's kind of an indication that it'll be important for its function, and we can target this when creating treatments. So kind of to support that information, I've also used this PFAM database and BLAST genetic research. So what PFAM does is it categorizes protein into its families and domains, which can give me an indication of other proteins that it might be similar to, and sort of the treatments that have targeted that and how it can apply it to this protein as well. And then finally, for the section where I'm looking at conserved residues, I was able to create a phylogenetic tree, which is a diagram demonstrating the evolutionary pathways among multiple species. So in the kind of the figure, there is a physical demonstration of the amino acid sequences across multiple species and the similarities and differences between them. So determining the areas of protein sequence that may show mutation bias across a phylogenetic tree may help develop drugs, as I mentioned before. And as the HNOX domain, where heme binds, or as heme binds to the HNOX domain and it's conserved, that is a kind of a good domain to target. Okay, so in order to generate more information to target SGC properly, it is imperative to ensure that hemoglobin and SGC do not have similar similarities, as they both have that heme binding site. So by studying interactions and the conservation of the protein, it is possible to dock the drug candidates and analyze the success using free binding values, toxicological properties, and a comparison to current FDA-approved drugs. So I use sequence alignment through residue pairing to see the similarities in sequence and structure between the proteins SGC and hemoglobin. And then finally, an analysis of binding affinities and RMSD, which is just the distance between different parts of the protein. When drug candidates are docked against a receptor, kind of demonstrates the effectiveness of a possible candidate. So I used Autodoc Vena and Chimera because it is quite accurate in its binding modes compared to some other options. And Chimera also gives us opportunity to look at protein ligand interactions based on the docking, which were analyzed afterwards. Through screening ligands, it's possible to determine which ligands have the highest delta G values and have good binding affinities. So the the figure demonstrated is a heat map of the different candidates, so that I screened, and sort of the highest values and the lowest values. So yeah, in the figure, there's some candidates, there's about 10 that have high potential, and 3, so C15H24 N4O, C9H11NO, and C16H20 N404 that had the highest binding affinities. So these drug targets can be identified, can move on to lab testing on animal models to be developed further into medications, as there is a lack of FDA approved treatments. And then finally, a combination of invasive treatment and medication can be used to reduce the chances of stroke or high blood pressure. So thank you for listening. The last couple figures are just some of the references I used. And yeah, thank you.
Thank you so much, Nambita. Fascinating project. Alyson, you're up.
All right. Okay, I'm going to share my screen now. Hopefully you can see it. Does this work?
Yes, looks great.
Okay, cool. All right. So hi, everyone. My name is Alyson, and I'm here today to show my research project on specific phobias in children, its etiology, treatment, and directions for future research. So first, I'd like to give a little background on why I decided to choose this topic. So it all started on February 25th, 2012 in Cancun, Mexico, and I was at the beach. And where there are beaches, there are a lot of birds. So here I was, this is an actual picture of that exact day. And essentially I was attacked by birds. So like 10 to 15 seagulls like all flew at me, and they all wanted a peck of my lunch. So ever since this day, I have been super scared of birds. Like walking down the street, if I see a bird in front of me, I will actively go to the other side to avoid it. Or if I'm like looking outside the window and there's a bird right there, I'll actually flinch and run away. So I wanted to understand how my brain works when I'm afraid of something and why this developed. So here is a little overview of what I'm going to be going over, and like a brief understanding of my project. So first, specific phobia is the marked fear about a specific object or situation that provokes an immediate anxiety and is actively avoided. So there are 4 types of phobias. First is blood injury injection. So this is things such as being scared of needles or fainting at the sight of blood. Next is nature environment. So this is being scared of things such as thunder or the rain. Then there's animal, which is like mine. And then finally there's situational, which is being afraid of flying in an airplane or riding a car. So specific phobia, especially in children, is one of the most prevalent childhood anxiety disorders, and it affects 5% of children in community samples, which is when researchers just take a general consensus of a community, and 19.3 adolescents in America. So there's a very limited understanding of specific phobia, especially in youth. So it's really important to study it to try to get the most effective treatments. So upon researching the neuroanatomy and neural responses of individuals with specific phobia, I found that the amygdala is one of the most important brain regions within this whole process, and it changes quite a lot in individuals with specific phobia. So here's the amygdala, and essentially the amygdala is responsible for the acquisition and expression of fear conditioning. So there's 3 nuclei within the amygdala. And when someone experiences a fear, this stimulus is sent through the 3 nuclei, and they work together to produce a behavioral manifestation of this fear response. So this can be things such as heavy breathing, dilated pupils, or a faster heart rate. And in individuals with specific phobia, there has been seen of a hyperactivation of this amygdala. So this means that there is heightened activity in the amygdala, and this takes place across all phobia subtypes. Next is the MPFC, or medial prefrontal cortex. So the medial prefrontal cortex is responsible for regulating fear behavior by modulating the activity of other brain regions. And through my research, I found that the ventromedial prefrontal cortex in particular acts quite differently. So in contrast to the amygdala, the VMPFC is hypo-responsive, which means there's diminished activity and it's quite suppressed when someone experiences a fear. And this takes place across all phobia subtypes as well. So the brain regions that I just talked about undergo various developmental changes throughout childhood and adolescence. So first, in the amygdala, it changes in its neurocircuitry of threat processing, which means the right side of the amygdala is actually increasingly sensitized during use. So this can be shown through how children and adolescents, it's quite more difficult for them to distinguish the difference between a threat and safe stimuli, whereas it's more easy for adults to do so. So this hypersensitivity actually puts them at a greater risk of developing specific phobia. The MPFC, on the other hand, it actually continues to develop postnatally or after birth. So it's not fully activated during childhood and adolescence. So there's very limited research in their relationship between the MPFC and the amygdala right now. So throughout my whole research, I found that familial origins was one of the most impactful and influential variables in the development and maintenance of specific phobia, especially in youth. So I found that parenting practices such as hypervigilance and overprotection, they actually kind of restrict the child's actions, thoughts, emotions, and it promotes the child to be more dependent on the parent. So when the child sees a parent being more heightened and they monitor their environment for cues that impair the safety, the child will actually imitate the parent and become more cautious and uncertain. Another parenting practice is accommodation. So this is when parents or families sort of alter their behavior to minimize or reduce their child's distress when they're exposed to the feared stimuli. For example, a child with nyctophobia or fear of the dark, their family might all sleep before the sun sets just to prevent their child from experiencing any discomfort. So even though this has good intentions, it's actually connected with more severe symptoms and poorer treatment outcomes. And it's also very consistent with the overprotection and hypervigilant parenting practices. So next, I'll be going over some current treatments within the field. So the first treatment is traditional exposure therapy. So this is when the patient and the doctor go through the patient's fear hierarchy. So this is an example of a fear hierarchy of someone who's afraid of spiders. And essentially the patient just works their way up the fear hierarchy until they're no longer scared of that situation. And then they'll move on to the next one. And the results have been relatively successful. However, patients are subject to up to 12 weeks of being exposed to the stimuli, which can be very uncomfortable, especially if you're near something that you're really scared of for such a long time. So this actually lowers the percentage of treatment completers and their discomfort just increases. That's why the popularity of OST or one session treatment has been very important. So this treatment lasts 1 day and takes place in only 3 hours, and it has these 6 steps over here. And it has been generally successful across all phobia subtypes, especially because it can simultaneously reduce the untreated phobic and anxiety disorders in addition to the targeted phobia. And it's also more suitable for families with less time and motivation for the longer term sessions. So here are a few studies that have demonstrated success in using one session treatments in youth. The next treatment I'll be going over is virtual reality. So sometimes in vivo therapy or like in-person therapy might not be the best choice because a lot of patients just don't feel comfortable being near their feared stimuli. Also, it might be difficult to get the resources to do the experiment. For example, if you're afraid of flying in an airplane, it can be really hard to get an airplane for 12 weeks and work up your fear hierarchy that way. So that's why virtual reality has been working to improve accessibility, affordability, and efficacy of the treatment. And it has been highly effective as well, as shown with these studies. And of course, there are various limitations in the field. So when I researched many of my papers, a lot of the participants were usually Caucasian and middle class. And treatments based on these studies may not work for diverse backgrounds. This is because people from other backgrounds and ethnicities, they have different parenting styles and different living accommodations, which can affect the prevalence of certain phobias. And like I said at the beginning of the presentation, there's also a very limited understanding of specific phobia in youth. And finally, there are various ethical limitations to this as well. Because fear learning paradigms evoke distress, there is a gray area in exposure-based therapy regarding consent, especially in children, because usually it's the parents making these decisions and the children don't really have any say. So in the future, a lot more research is required to better understand the amygdala and MPFC relationship during development and in youth, and new imaging techniques are working through this right now. And a lot of the studies that I read, they have been looking into the effects of other caregivers on phobia development. So this can be things such as a teacher, daycare provider, and other relatives as well. Also, many studies have been adjusting OST and virtual reality treatment to try to find better and more effective ways to do so. And finally, just like I said in the limitation slide, there must be different metrics of diversity to be explored because we can't understand the full extent of this disorder until we do so. All right, so that's the end of my presentation. Thank you everyone for listening, and I hope you enjoyed it.
Thank you so much. That was great. Your slides were very well put together and with nice animations. Saanvi, it's the floor is yours.
Fantastic. All right, can everyone see my screen?
Yes.
Great, okay. So hello everyone, my name is Saanvi, and my research project is about how using cranial radiation therapy to treat a cancer or tumor can cause consequential genetic alterations that lead to a tumor regrowth years later. So to preface the presentation, I'll be going over these ideas in order. I'll start by giving background information on what gliomas are, and then move on to what radiation therapy is and how it is used to treat cranial issues. From there, I'll transition to how side effects of radiation therapy lead to the development of a glioma. I'll wrap it up with the conclusion and considerations of the information compiled in my presentation. Gliomas are the most common and malignant neurological growth, responsible for around 30% of all brain and other CNS cancers in adults. Gliomas have low survival rates and poor prognosis, meaning the life expectancies of glioma patients are not favorable. Here's the image you can refer to. It has specific examples for gliomas, but it applies to all different types of tumors. A glioma is categorized as an astrocytoma, oligodendroglioma, or oligoastrocytoma based on its genetic profiling and histology, which is the structure of a tumor tissue. As the image shows, the World Health Organization, or WHO, further classifies tumors on a scale of 1 to 4 based on how aggressive the tumor is or how proliferative it is. We use the word proliferation to describe how rapidly the tumor spreads and develops. The idea of proliferation is very important to understand how radiation-induced gliomas first develop. So we'll continue to see this word throughout the presentation. If a tumor is less aggressive and develops slowly, it'll be classified as a low grade, such as 1 or 2. But if a glioma such as glioblastoma is more malignant, meaning it spreads quicker and has a worse prognosis, it is classified as grade 3 or 4. And one thing I would like to mention is that this chart is slightly outdated, as the WHO started using Arabic numerals instead of Roman numerals, but it is still very relevant. So radiation therapy is a countermeasure for various tumors. There are 3 types of radiation therapy: systemic, internal, and external. Systemic radiation is when a radioactive substance is either swallowed by the patient or administered through an injection. Internal radiation, also known as brachytherapy, is when a metallic substance that has a radioactive seed is surgically placed inside or near the tumor. Last but not least, external radiation, the most common option, is very non-invasive and is used by using machinery to regulate the transfer of high energy electromagnetic waves from outside the body to within the tumor. In simplest terms, radiation therapy works by creating DNA double-strand breaks, or DSB for short. This damages the DNA in cancer cells to stop them from further replicating. Then DNA damage responses, or DDRs, occur to repair normal tissues that may have been harmed. Medulloblastoma represents about 30% of pediatric brain tumors and 7 to 10% of all brain tumors. The etiology, or specific cause of medulloblastoma, is unknown, but there's conclusive information about its progression and treatment. Primarily, neurosurgery is used to resect or remove part or all of the tumor. In many cases, radiation therapy is used prior to the surgery to shrink the tumor or after the operation to reduce the risk of some remaining tumor cells duplicating to grow into another tumor. With over 6,500 new cases per year of acute lymphoblastic leukemia, It's a very serious issue that is mostly prevalent among children. ALL is a cancer of the blood and bone marrow due to irregularities in the DNA of marrow cells. It has a high proliferation, meaning it spreads rapidly, and it can be fatal within a matter of months if left untreated. Chemotherapy is a common treatment for leukemia by using powerful chemicals to kill leukemia cells. However, in situations where chemotherapy has not been effective, radiation therapy is the next best alternative. Radiation therapy is also used in instances where leukemia has escalated to the brain or spinal fluid. This is where ALL patients are exposed to cranial radiation that can cause some patients to develop a glioma years later. So up till now, we've discussed how radiation therapy is used to treat ailments, and now I'll transition into how radiation therapy can have negative effects after treatment. A study observing the after effects of radiation therapy found an association between the therapy and secondary tumor growth due to genetic alterations. The 3 genetic alterations I'll describe specifically cause cells to proliferate too quickly. When this happens, new cells do not have important components, or they have genetic alterations in their DNA that are very consequential. These are defective cells, and they multiply into more defective cells, eventually forming tumor tissue. After radiation therapy, scientists observed a CDKN2A deletion. This gene provides instructions to create tumor-suppressing proteins, so the deletion of this gene leaves cell proliferation unfacilitated, and proliferation is too rapid. Another alteration is the amplification of a gene called PDGFRA, which is short for platelet-derived growth factor receptor alpha. PDGFRA plays an important role in organ development and wound healing, and it activates the signaling pathways that facilitate normal, healthy glial cell mitosis. So in another study, scientists concluded that PDGFRA amplification negatively impacted prognosis or survival rates for tumor patients. The gene EGFR is also impacted similarly to how PDGFRA is, and this amplification is seen in 40% of all gliomas, and its mutation also causes cell proliferation mechanism to perform too rapidly. So the main takeaway from these points is that radiation is used to damage the DNA of tumor cells, just like it does with medulloblastoma. However, it's not feasible to avoid damaging a normal cell because of how incredibly tiny they are. So modern radiotherapy techniques are not accurate enough to only target the malignant cells. So then healthy cells that do get targeted sometimes become defective, and this defect spreads and grows so much that it creates another brain tumor or a glioma. This is what we call radiation-induced glioma because the genetic alterations are a consequence of radiation therapy. So considering that the latency period between primary malignancies and radiation-induced gliomas ranges from 2.5 to 35 years, the studies are clearly not based on the latest radiation techniques, leaving room to question if modern radiotherapy still has the same risks. Over the last 2 decades, scientists and engineers have been developing more precise procedures and devices, such as dose conformity tools, that are able to administer radiation more precisely to the target and reducing the impact on normal tissues. Radiation therapy significantly impacts quality of life, either positively or negatively, sometimes both. Radiation therapy is commonly used as an aid to slow the spread of cancer's growth, or used in conjunction with chemotherapy and surgery to remove the tumor completely. However, radiotherapy also comes with the risk of impairing patients' lives after treatment. Radiation-induced gliomas display multiple genetic alterations caused by radiation treatment. This information should be reviewed by patients when considering radiation therapy. For some, an alternative treatment with less risks may be appealing. For example, targeted therapies, which are not widely available on the market yet, may become more popular alternatives. To continue striving to improve patients' quality of life, it's imperative to continue supporting research for developing minimal risk radiation therapy mechanisms and to monitor patients for the appearance of secondary malignancies. Thank you.
Thank you so much, Saanvi. It's a very thorough project. And then last but not least, we have Angie.
Okay, I'm gonna try to share my screen. You guys able to see that?
Yep, looks great.
Okay, cool. Hi, I'm Angie, and my project is about biomimetic blood cell coated nanoparticles for improved site specificity and enhanced bioavailability. Yeah, so what are nanoparticles anyway? Nanoparticles are small molecules or atoms with at least one dimension in the 1 to 100 nanometer range. And this means that nanoparticles are really very small. And this allows them to have a very high surface area to volume ratio. This means that they are also very highly tunable. If you change the material that makes the core of the nanoparticle or change the material that makes the coating of the nanoparticle, you can change different properties like the charge and what type of materials it is attracted to. Nanoparticles are created with two main methods, the top-down and the bottom-up approach. So with the top-down method, you take bulk material and you break it down into smaller particles to create your final nanoparticle. And for your bottom-up approach, you take very, very small molecules and you synthesize it to create your final nanoparticle. And nanoparticles actually have many product applications in our daily life. This includes things like solar cells and paints to prevent deterioration, and in medicine, which is what I decided to focus on. So in medicine, nanoparticles actually have many different advantages and I guess it creates sustained drug circulation lifetimes, reduced side effects, and site-specific targeting. So nanoparticles in the body actually do have many barriers, and one of these barriers is the immune system. So our immune system has macrophages, and these macrophages basically clear any foreign material in the body, and this is usually things like unwanted viruses or bad bacteria. But in this case, when we introduce a nanoparticle with a helpful drug into the body, the macrophages will also clear this nanoparticle, which is not something we want. So scientists have tried many different ways to sort of combat this problem. This includes cancer cell membrane coating, which is made to target cancer tumors, pegylation, which is used to avoid macrophages as a whole, blood cell coating, which I will talk about later, virus protein shells, which are made to target cells the virus used to attack, and self-peptides, which are sort of made to trick the body into thinking that the nanoparticle is supposed to be part of the body. And so for my project, I decided to focus on blood cell coating because I thought it was a very promising method of biomimicry, and it's a very interesting way of attempting to target these tumors for nanoparticle drug delivery. And so blood cell coating for nanoparticles are done with two main methods, the carrier and the camouflage system. And so with the carrier method, you're actually taking physical blood cells and you're putting them in hypotonic solution and lysing it. So the insides of the blood cell are taken out, and then you inject the nanoparticle with your desired drug into the blood cell. So you're directly taking the blood cell and putting your nanoparticle back in. With the camouflage system, it's slightly different because you're taking the proteins of the blood cell and then you're sticking it onto the coating of the nanoparticle. So this is indirectly using the blood cell in order to coat your nanoparticle to make it more efficient in the body. So one specific way this is a type of camouflage system is using self-peptides. So CD47 is one of the most abundant proteins in red blood cells. So you can harvest the CD47 and attach it to the nanoparticle. Here they are referred to as nanobeads. And when you attach it to the nanoparticle and introduce it to the body, this is able to increase circulation and sustain the lifetime of the drug in the body and allow it to avoid macrophage clearance, which is what we want in these nanoparticle mediated drugs. So as I mentioned before, with the red blood cell coatings, you're able to further target different parts of the body in order to target these diseases. So one example is using serum albumin, and serum albumin is another protein in the blood. And serum albumin accumulates in tumors for different reasons, and it's overexpressed. So if you coat a nanoparticle with the serum albumin, it will be attracted to the tumor and therefore increase the site specificity of the nanoparticle targeting. Another example of this targeted therapy is reactive oxygen species, also known as ROS. And how this works is you take the red blood cell and you coat the nanoparticle, and this creates an ROS nanoparticle. And once it's introduced to the body, it directs to a site and then it oxidates to release all the drugs. And this is able to increase site specificity and make sure that the drugs are going to the right place and decrease side effects. So with chemotherapy, I'm sure we all know there are many side effects, and nanoparticles are a way of trying to mitigate these side effects. However, with the ROS, sometimes there are complications because of hypoxia in tumors. So hypoxia is essentially where tumors lack oxygen. But one way to combat this is using nanoparticles and filling them with perfluorocarbons. And perfluorocarbons are they have high oxygen solubility. And because they have high oxygen solubility, it's able to act as an artificial blood substitute and oxygenate the tumor area, and therefore allow the ROS species to oxidate and be more effective. As I mentioned before, nanoparticles are extremely tunable, and the same can be said with blood cell coated nanoparticles. And one thing you can change is the different protein to lipid ratio on the membrane. So the more membrane proteins, the more white proteins from the white blood cell, it's able to lead to higher efficacy of the nanoparticle in the body. Again, this can be seen with CD47, which we discussed before. Compared to low expression, high expression CD47 leads to higher success of avoiding macrophage clearance. Another interesting aspect of nanoparticles is that we may think of them as spheres, but they can also come in many different shapes. So in another study, it was found that nanoparticles that were designed to be non-spherical and had red blood cell membrane coating creates a reduction in uptake by the immune system. So the combination of creating a non-spherical nanoparticle and having the red blood cell membrane coating is able to effectively stop the macrophage clearance. Thank you for listening.
Okay, so thank you all to all the students for your hard work and really truly excellent presentations, very impressive work. Now I'm going to open it up to Q&A. So if you have a question, if you're one of our attendees or one of the students, you have a question for another student, you can go ahead and type a question into the Q&A and I will address it. I'm not seeing anything there now, so I'll encourage you to think of something, but in the meantime, I'm happy to ask some questions myself. I'd like to start with Nambita. I'm really curious, like, what the origin of your project is. Like, where did you get the idea, and what sort of drove you to that particular topic?
Yeah, so actually the disorder I was talking about, moya moya, it was something that a close family member of mine had. And, as I was, when I'm researching this disorder, when we first found out, and I was kind of surprised at sort of the lack of, it being addressed and information. So I kind of went on this rabbit hole of looking at this protein, and then I thought to myself that I could potentially apply the software and, kind of solve the questions that I was trying to look for. Yeah.
Yeah, that's so interesting. Speaking of personal experiences leading to inquiries, Alyson, I love how you wove in this personal story about what led you to your particular topic. I was curious, like after you went through all your research, what did you learn about yourself, and did you apply any of the techniques that you learned about?
Well, at the end, I realized that maybe treatment is not for me because I can't even like looking at a picture of I will physically use my hand to cover up the picture because I just can't look at. So virtual reality is a possible opportunity for me. However, right now I think I'll just stick to avoiding birds. But it was really interesting to really deep dive and find out what exactly goes on in my brain when I see something that I'm scared of. And it wasn't something that I had known a lot about before. So I was really surprised to see all the treatments being done and a lot of the studies that have been focusing on youth especially.
Yeah, I have a little one. She's 8 now, and she has a lot of phobias. So when you were talking about the caregiver experience and accommodating and things, it really kind of struck me like that I should look up and learn more like research of ways I can better support her versus just trying to protect her from the things I know she's afraid of. So it was a really fascinating literature review. So thanks for sharing. Let's see. Saanvi, I have, similarly curious, like where people get their ideas for projects. Is it something that was personal for you, or like where did you come up with your topic idea?
Yeah, so it's personal. To begin with, I'm interested in neuroscience because I have a family member with autism, which is kind of what introduced me to more of it was more of a psychology introduction and understanding that, how people think differently and how differently they respond to certain things. And I also had, I lost a family member to cancer. So I wanted to do research into cancer treatment or tumor treatment. And I found why not combine neuroscience and also cancer treatment. And so I started just looking around and finding some interesting articles. And I came across this very interesting thing, radiation-induced glioblastoma. And I found it peculiar that treatment for one tumor can actually lead to another tumor. And I wanted to look into it a lot more in depth. So that's how I did research on this.
Mm-hmm. Yeah, it was great. Very interesting. Angie, for your project, I was curious, like maybe a surprise that you had, something you didn't know as you were going about learning. Like, do you have an example you can share?
Yeah, I think something that I came across, I did a lot of general research on nanoparticles as a whole, not just the blood cell membrane coating. And something I found that is actually going through testing or something is using magnetic fields with these nanoparticles. And I think that was kind of crazy to me because you can externally manipulate these nanoparticles to go to like where you want in the body. And that is something I want to look more into in the future, and something I found really interesting as I was researching for my paper.
Mm-hmm. Yeah, awesome. Another area that I was curious to hear more from all of you guys about is, as a mentor myself, I know that it's kind of it can be a rocky road, like as you're going through your independent research project. I was curious to hear from some of you about the most challenging moment you had. I guess I'll ask Alyson that question.
Yeah, so I think for me, one of the most challenging aspects of this research was finding the neurobiological origins. So I knew from the beginning that I wanted to have even though it was more of a psychology focused paper, I wanted to have a little bit of background on neuroscience and what goes on in the brain. And there was definitely not very much information on that. And I had to go through countless papers on like all of these topics. And I found that a lot of phobia was linked to other anxiety disorders. So it's like comorbid. So a lot of anxiety, a lot of neural responses to people with anxiety are also very similar to those in specific phobia. However, the brain regions such as the MPFC, it acts quite differently because it's actually like there's a lot less activity seen in this area than in the other disorders. So that was quite interesting to do. And yeah, I think that was one of the most difficult parts. And then another difficult thing I would say is looking at limitations. So I don't know if you remember, but in my paper, I talked about like a lack of diversity within the sample group. So it was hard to find treatments that would appeal or like would be good for someone who looks like me. It was mostly children and people from upper middle class and Caucasian households, and they tend to have different parenting practices. So it was a little bit difficult to sort of accommodate that to what I was looking for, but I did find some studies that helped, and overall it was smooth sailing after that. But yeah, those were the few challenging aspects of mine.
That's so interesting. Yeah, I love that part that you brought up because I think so often when you look at like traditional neuroscience and stuff, like they look at the brain and it's like the brain only, and they're not thinking about the cultural conditions that brain is living in, right? We're human beings and we have family around. And so that is really fascinating that parenting style or cultural practices and things like that can really impact how effective certain treatments are going to be, particularly when they need to be sort of supported by, when you have a young child that has caregivers and stuff. So everybody sort of needs to participate and be on the same page. So really, really nice angle as you're bringing into the project there.
One thing that was quite interesting that I didn't add in my presentation was a lot of the times, even I tell myself, oh, just face your fear, just face your fear. And I learned that term actually sort of holds children back and actually causes them to be more cautious and not as confident when they're looking at the thing that they're scared of. So that was an interesting thing. Yeah.
That's great. Saanvi, what about you? What was one of the more challenging elements that you had during the course of your project?
It was actually, Alyson mentioned at the beginning that she couldn't find enough information, but for me, it was the opposite, where initially it was hard to figure out what I wanted to look for, but when I found out, there was so much information available and I had trouble pinpointing what aspects do I want to focus on? And that kept changing throughout my project journey. So it started off with, I just want to focus on glioblastoma, but then I started venturing more into the primary malignancies and ended up with what I presented today. And another challenge could be that there's not enough information about specifically how modern radiation therapy is different from how it used to be a few years ago or a few decades ago. And that was one of the things mentioned in my considerations. And I wish I could have found more information about that to talk about, because I feel it's very relevant.
Yeah. Sometimes you learn things when doing these literature reviews that could point you in a direction of like research that needs to be done. And when you're in a place where that's possible to do, like as you move into your further into your educational career and to college and things like that, like it gives you an opportunity to carve new paths yourself. So hopefully doing these reviews is a good foundation for all of you if you continue on your journey. Nambita, what was a big challenge for you?
I mean, I think other than all the, whenever you're working with stuff, there's always issues with that and the code. But I think just generally, I mean, I started off with quite a large general idea of, I just want to know more information about this protein and how we could target it. So going from that and figuring out, okay, so what do we need to do first? So like, maybe I should find a model, and then I look online and there aren't very accurate models, so I have to sort of create one myself. So kind of constructing that step-by-step process was at first slightly challenging, but I think it did also make it kind of engaging and fun in a way. But when I was able to complete it all, yeah, congratulations.
And then Angie, what did I don't think I asked you yet, but like what was a big challenge for you and your project?
Yeah, I think I was kind of similar to Saanvi. Like it was kind of hard to narrow it down. There was so much information, and I finally settled on like membrane coding and things like that. And I think something else that was kind of difficult for me is sort of like visualizing what nanoparticles actually do like in the body. And I think I did have to go around, find a lot of clinical trials. Like I wanted to know more about like what they were actually doing in the field and how they're actually applied. And I think that was one of the more difficult parts for me, and like just trying to truly understand what they're able to do.
Yeah. Well, anyway, I would like to say that I'm humbled and I'm really impressed by all of the work that you guys have done, and congratulations to you. And I wish you the absolute best of success as you move through your career, but you obviously all have tons of potential. I've had a blast learning more about your projects today. I hope you've enjoyed hearing from each other as well. So I'm going to go ahead and close this session with a round of applause to all of our scholars. And thank you for being here today. Have a good rest of your Saturday, everyone. Bye-bye.
Bye-bye.
Thank you.